Hello everyone, my name is Drew Voigt and I'm with the University of Iowa Institute for Vision Research, and in this tutorial video I'm going to show you how to use all of the functionalities within Spectacle, which is an interactive resource for exploring single-cell gene expression studies in the eye. To access Spectacle, simply open any web browser and navigate to https://singlecell-i.org. As of July 2021, we've updated Spectacle and introduced several new features. So if you're a returning Spectacle user, we've provided a few bookmarks in this video so that you can skip around and learn about features that are of most interest to you. So when you navigate to Spectacle, this is the landing page, and you'll notice a variety of menu items on the left-hand side. The first menu item that's automatically selected is the dashboard. So the dashboard presents the user interface that will be filled in with our interactive plots once we load a data set here. But we also have a few other menu items of interest. The first of which is this tab called Datasets and Citation Information. So this tab has links to all of the different papers that describe the different data sets provided in Spectacle, and you can read these papers to learn more about the study design and included donors for each of these data sets, as well as download the R data files for all of the data on Spectacle. Below that, we have a tab called About and Contact. This provides a little bit of information about our team at the Institute of Vision Research and a contact email address for any questions or comments. We also have a how-to guide that provides a link to this video overview, as well as the detailed written guides to accessing different features of Spectacle. And then the last tab I'll talk about is this changelog. And this changelog is going to be filled in with different updates and changes to Spectacle and how we'll communicate those changes with you. So that's a quick overview of the Spectacle interface. And now I would like to go over how we can open a data set and start exploring gene expression. All right, now we're ready to load a data set within Spectacle. In order to load a data set, we must first navigate to this top dashboard menu item, viewing the landing page of Spectacle, and then we can select from the drop-down menu one of 7 currently available data sets. And each of those are briefly described on the right-hand menu, this quick start guide, and in a little bit more detail in the datasets and citation tab. So for this first part of the tutorial, I'm going to load the dataset Retina, Fovea versus Periphery, which contains paired foveal and peripheral libraries from 3 human donors consisting of neural retina. The first image that appears is the dimensionality reduction plot. Each dot on this plot represents the multidimensional transcriptomic profile of an individual cell, and cells are clustered into groups of cells with similar gene expression profiles, which represent likely shared cell types. This plot is made fully interactive with the R package Plotly, and you can hover over cells and view individual characteristics. So for example, this cell I'm hovering over is a glial cell from the periphery of donor 3 and belongs to cluster 13, which you can see in this pop-up. With Plotly, you can zoom in and view a more granular view of the data by just simply dragging and dropping your cursor. You can double tap or hit the auto scale button to zoom back out. Plotly also enables you to download the plot as a PNG file by clicking this camera icon. Simply click the icon and you can download a plot to your downloads folder. With this visualization, we see the different clusters of cells which were categorized by our manuscript, but we can also view different aspects of metadata or biological variables from this study. So from this dimensionality reduction plot color drop-down menu, we can view cells colored by their library. So here in shades of red, we see foveal cells, and in shades of blue, we see cells originating from the periphery. And we can also view different biological variables. So in this example, it's just another visualization here. In red is foveal cells, and in blue is peripheral cells. In different studies, we have different biological variables that can be used to visualize, and these include things like region that we acquired the cells from, or disease status, or genotype. So now that we've loaded a data set, The next tool that we want to overview is how to explore gene expression across these cells. A major use of spectacle is to identify what cell population expresses a gene of interest. And there are a couple of really good ways to do this. The first of which is to use the heat map tab. If you click on the heat map tab, you can scroll down and type in the gene name that you wish to visualize the expression of. So for this example, I'm interested in the cone-specific gene arrestin-3, or ARR3. After hitting enter, cells in the cartoon are colored based on their expression of arrestin-3, and the color of each cell type in the cartoon corresponds to the average expression of this gene across all cells in the given population. So we can see here that the cone photoreceptor cells, colored in dark blue, express high levels of arrestin-3, while most other cell populations don't express any of this gene. We can change the color of the expression scale by clicking within this heat map color box, and we can change this to green or any other color if we like. And this can be really useful if you're preparing a presentation and you want to match a color theme, or if you want to pair this expression information to an immunohistochemistry image. If we'd like a more granular view of the data, we can turn off this display heat map as a cartoon slider. And then we can visualize gene expression across each cell individually. I'm just going to change this away from green to a warmer color here, maybe even this darker red. So we can hover over information from each cell and view at the bottom of this hover window the actual log expression level of this gene across each cell. Sometimes, though, it's useful to visualize co-expression of two different genes. So to do this, we must turn on the Visualize Co-expression slider bar and enter the second gene name. So here I'm interested in co-expression between arrestin-3 and the cone-specific phosphodiesterase PDE6H. After hitting enter, the co-expression plot appears. And we can see that on the x-axis we have the expression of arrestin-3 in gray to red, and then on the y-axis we have expression of PDE6H in gray to blue. If we zoom in on this plot here, we can see that many cells co-express both of these genes in this dark purple color. We can contrast this with comparing expression of arrestin-3 and the rod-specific gene, phosphodiesterase 6 alpha. We can see here that the cone photoreceptor cells in red express lots of arrestin-3, and the rod photoreceptor cells in blue express lots of PDE6A. So the last way to overview gene expression is with the violin plot to the right. And this violin plot enables us to look at expression of multiple genes at the level of each cell population. So we can again enter our cone-specific gene arrestin-3 and any other genes that we wish to visualize. So I'll look at PD6 alpha, and we can see that the arrestin-3 gene is expressed by our cone populations, and PDE6 alpha is expressed by the rod populations. The neat thing about this plot is we can query as many genes as we want simultaneously and view this stacked expression at the population level. Spectacle also supports reclustering, which allows for potential subgroups of cells to be identified. So in this example, I've loaded a different data set, this Retina autoimmune retinopathy versus control data set. And when we load this data set, we see that there are three different populations of bipolar cells in clusters 17, 18, and 19, which broadly correspond to cone on, cone off, and rod bipolar cell subtypes. There have been many studies that show that there are several different populations and subpopulations of each of these subtypes, and so we want to identify if we can see gene expression features of any of these. So we can hit the Recluster tab and select the groups we want to cluster, which are clusters 17, 18, and 19. We can hit the Start Reclustering button, and we are prompted with a warning that this reclustering is slow. And that's because a lot of this math that is done in reclustering is very memory intensive. The expression matrix for these specific cells need to be renormalized and rescaled, and then dimensionality reduction needs to be recomputed for several different methods for these populations that are selected. So in general, I recommend selecting only a few thousand cells for reclustering. Otherwise, the server can actually require so much memory that you receive a crash. What's nice is a progress bar appears on the bottom of the window, telling you at what point in this reclustering process you're at. So here we can see that we're just running the UMAP dimensionality reduction, and we're almost done with the reclustering. But this can take anywhere from 30 seconds to 1 to 2 minutes, depending on how many cells you select. So as we wait for reclustering to finish, we'll scroll up to the top of this window, and in this region is where a reclustered plot is going to populate. All right, we've finished. And we can now see that these bipolar cell clusters, which kind of correspond to this tree-like branch here, when they're reclustered, they have this star-like pattern. And we can see lots of different subclusters. So we can color these cells in several different ways. Here we've colored the cells by their original clusters, and we can see where clusters 17, 18, and 19 corresponded. But we can also color cells by a new cluster label. So here we've broken this into 21 distinct clusters, or by their original library. So here we have, in shades of blue, some foveal, shades of green, peripheral, and then red corresponds to this donor with autoimmune retinopathy. Or by other characteristics like disease status, no in red, and autoimmune retinopathy in blue. But for this visualization, what's going to be really useful is coloring by a gene. And so one gene that marks a bipolar cell population of interest is RELN. So when I enter in RELN, I can see that this subpopulation of cells right here, towards the top of this reclustered plot, very strongly and specifically expresses the RELN gene. I can contrast this to our original dimensionality reduction plot, RELN, and I can see that there's not really a subpopulation within our original image that expresses this gene. Another gene that demonstrates interesting subpopulation expression is ERBB4. So ERBB4 marks a specific class of cone-off bipolar cells called the BC3A subclass. And again, we see a really strong and specific expression in one small subcluster here within the reclustered object. But in our original object, we don't really see localization of this cell population within the original cone-off bipolar cell cluster. So really this reclustering function can be powerful for identifying small cell populations of interest. But again, there are some computational and memory limitations, so I encourage the user to be judicious when selecting the number of cells to recluster. Spectacle also facilitates very flexible differential expression analysis. So for this example, I'm again going to use the retinal autoimmune retinopathy versus control dataset. In order to access the differential expression features, simply select the differential expression tabs on the top. If only one differential expression tab is selected, a text box warning will prompt you to select the second differential expression tab as well. When both tabs are selected, this dimensionality reduction plot is displayed. The first decision the user must make is what comparison will be made with differential expression. So I want to initially compare gene expression between cluster 5, which are Mueller cells from the autoimmune retinopathy donor, versus clusters 7 through 10, which are Mueller cells from our control donor. By default, differential expression is performed at the cluster level. So I select group 5 versus group 7, 8, 9, and 10, and I can hit Start Differential Expression Analysis. We get the status bar of the differential expression status at the bottom of the screen, and while that analysis is running, I'm going to explain a couple of the key settings for differential expression. The first of which is this log fold change threshold slider bar. So the log fold change slider bar will only perform differential expression on genes with at least a log fold change difference of the specified value. Likewise, the minimum percent threshold will only analyze genes that are expressed in at least this proportion of selected cells. So for this example, only genes expressed in greater than 50% of cells in one of our two populations will be included in this analysis. Setting these thresholds to higher limits results in much faster computation times, but is less sensitive. Conversely, setting lower limits to these thresholds identifies more differentially expressed genes, but is much slower. Here, I left the thresholds at their default values for spectacle. We can see that the analysis is completed because these graphical and tabular results have been populated. I find this first plot very useful, but it takes a little bit of getting used to. Here, each dot represents the differential expression results of a single gene that can be hovered over. The y-axis depicts our log fold change. So positive values represent genes enriched in our group 1, and negative values represent genes enriched in group 2. And we get a reminder of what our group 1 and group 2 selections are in this plot to the right. We can see that our group 1 is this autoimmune retinopathy mular cell population. The x-axis of this plot is a variable called delta percent, and this communicates the proportion of cells in group 1 that express each gene minus the proportion of cells that are expressed in group 2. So for example, I'm going to sort this table by genes that are enriched in our group 1 or autoimmune retinopathy selection. We can see that our most enriched autoimmune retinopathy gene is GFAP, and it's expressed by 98.5% of cells in group 1, but only 39.5% of cells in our control Mueller cell group 2. Therefore, this delta percent is going to be just under 60%, which we can see when we hover over this gene. This means that about 60% more cells in the autoimmune retinopathy cluster express GFAP. So this is really handy because this plot communicates the absolute expression change on the y-axis and also the proportion of cells expressing each gene and that enrichment along the x-axis. This tabular format of the differential expression results, you can search for individual genes with the search bar or export this into a CSV or Excel format. Differential expression analysis can be even more flexible. So if we scroll up, we can narrow our cluster selections with the lasso tool. So by clicking this lasso tool selection, we receive a second plot on the left, and we can get really granular and say, well, we have cone cells here. I wonder what separates these cones in kind of this left branch from cones in this right branch. We can hit Start Differential Expression. Again, we receive this progress bar of differential expression status. And once that differential expression analysis is complete, that progress bar disappears, and we can then view the results in the same format as before. And then finally, and this is a very useful feature, we can perform differential expression between cells of different biological conditions. So for this example, I want to look at differential expression between bipolar cells of control and autoimmune retinopathy donors. And just to show you, if we color our cells by autoimmune retinopathy status, we can see that the bipolar cells, there's not clear clusters that discriminate these cells. And so what I want to do is do a lasso selection and perform differential expression between autoimmune retinopathy status. So I'm going to select one of our bipolar cell populations here and hit Start Differential Expression Analysis. And in this analysis, we will determine what cells are enriched in our autoimmune retinopathy versus control donors. So if we zoom in on this plot, we can see cells in blue are bipolar cells from the autoimmune retinopathy donor. Cells in red are from our control donors. And only a few genes pass our thresholds here, but we can hover over and explore these genes in the same formats as described above. The last feature of Spectacle that I'd like to overview is our Regional Expression tool, which can be accessed in the left-hand menu by clicking Regional Expression. Regional gene expression in the retina has been very high interest to the vision research community. our group and other groups have completed several single-cell RNA sequencing experiments that compare gene expression across multiple different regions of the human retina. This regional expression tool allows the user to compare central versus peripheral gene expression across 6 of these independent studies. At the top of this window, we can see a cartoon depicting the design of each of these 6 studies. For example, in our most recent study, we compared gene expression between the foveal and the directly surrounding parafoveal retina. Other previous studies have compared foveal or macular retina versus more peripheral retinal samples. To compare regional expression patterns of a gene, we first select the cell population of interest from the cell input dropdown. I'm going to scroll down and select Mueller cells for this example. Then, from the gene input dropdown, I'm going to select the gene that I'm interested in. So the first gene I'm going to explore is CYP26A1. After hitting enter, this bar plot visualization appears. Each bar represents the log fold change between the central and the most peripheral library for each study. And the bars are colored according to the study in this top overview cartoon. A positive log fold change indicates that the gene is more enriched in the centralmost library, and a negative log fold change indicates that the gene is enriched towards the peripheral retina. As we can see from this plot, CYP26A1 is highly expressed by the more central Mueller cells across each of the 6 studies. This enrichment is particularly interesting as Dr. Sepko has found that CYP26A1 is highly expressed in the high acuity area of the developing chick retina. We can contrast this expression enrichment to the gene transferrin, which is an iron binding protein. We can see that in most of these studies, transferrin is enriched more towards the peripheral retina, in contrasting this to the CYP26A1 expression. Overall, this tool allows the user to select for any cell population and any gene the regional expression enrichment across different regions of the human retina. In this tutorial, we overviewed how to load different retinal, RPE, and choroid datasets, how to explore expression in heat maps and violin plots, how to recluster cells to identify distinct subpopulations, and how to perform differential expression between different biological variables or different cell populations. We also overviewed how to compare regional gene expression across 6 independent retinal human single-cell RNA sequencing studies. We hope that these tools aid you in your research endeavors, and please contact the Spectacle team at spectacle.iowa@gmail.com if you have any questions or comments about this tool. Thank you for your attention.